I hope you enjoyed the first part of the conference. I found it to be very, very interesting. We have two more speakers before the next question. Before we take in the next question break. Before I introduce the next speakers, I would just like to say to you that first of all, we would really appreciate if you could post the questions in the Q&A, because it's much easier for us to follow what's being asked if it's in the Q&A. And please keep your mics on mute. And if I forgot to mention anything else, Sandra, please feel free to jump in. The next speaker is Dr. Kьяртан, Herrik, and please correct me if I'm pronouncing it wrong. And he is a principal scientist at Lundbeck in the Department of Circuit of Biology, I think it's pronounced. And he will be talking a bit about the EEG and behavioral research data on pharmacological modulation of NMDA receptors, including GLUN2B selective modulation. Really interesting things. And the speaker after Dr. Kьяртан is Jenny-Li, and she's a mom of a child with another rare genetic disorder. And she's been a very, very, very active part of our GRIN research discussion group on Facebook and has a wealth of knowledge. And we are really looking forward to hearing from both of you. So Kьяртан, you can take over now.
Okay, thank you. I'll try to share my screen. It should come up now. Can you see my screen?
Yes, looks great.
Good. We'll need a pointer. Okay. Well, thank you so much for inviting me. It's a great honor to be part of this meeting. I'm really impressed by the talk so far. I've actually learned a lot, and I think they've helped me a lot because I can jump right into what I want to talk about. Yes. So my name is Kьяртан Hерrik, and I work at Lundbeck, and I will present some of the data that we have we've made at Lundbeck. It's both behavioral research and EEG. I have a little bit of both preclinical and clinical EEG that I would like to share with you. First of all, I think I need to put a disclaimer. So I do work at a company, a drug company. Our purpose is to restore brain health. That's very simple. Actually, it's really, really difficult, and we're fighting the best we can. But to my knowledge, we don't have a treatment option that we can offer GRIN patients, I'm afraid. But what I can is that I can tell you how we work at Lundbeck, how we work with the NMDA receptor, or examples of how we work. And as you may see in the background of me, I'm in Copenhagen. I work in the headquarters in Valby, but Lundbeck is all over the world. But the headquarters are here in Copenhagen. So I put down the questions that I thought were the most important to discuss, at least from my perspective. So first of all, I would like to tell you why I studied the NMDA receptor, because I come from a slightly different angle than you guys, I think. And I would like to stress how important the site of action is when modulating the NMDA receptor. And also why timing is of importance. And I would like to come into why this, or how the data that we have constructed translate to humans. So in the end, I think we can discuss how a brief inhibition of the NMDA receptor is relevant for GRIN patients. And I'm sorry, I have to throw out my son. I'm working from home as so many other people are. Excuse me for that. Okay. So I also made a drawing of the NMDA receptor, and I hope that you know it so well that I don't need to spend too much time on it. But what I want to stress again is how we came into this. I was really fascinated by the antidepressant effects that ketamine has been shown to have in treatment-resistant patients. And ketamine is blocking the NMDA receptor pore, just as other pore blockers you heard about memantine earlier today. And what ketamine is doing in these treatment-resistant patients is that it has a really rapid-acting antidepressant effect, but it's also inducing psychotomimetic side effects. So this is the effect of blocking the pore. There's also the proteal side that you've also seen before today. Here, this is selectively on NR2B subunits of the NMDA receptor, and there are some compounds that can negatively modulate the receptor through this site. They have also shown to have antidepressant efficacy, but not without the psychotomimetic side effects. We know less about these, so we don't know how they affect sleep, for instance, or motor behavior. And then, as you've seen before today, there's the glycine side where D-cycloserine is binding. Here, another type of compounds. One that I will mention today is D-cycloserine binds, and so D-cycloserine is a partial agonist. So that means that it is less effective as serine or glycine at the site. So in the way we study the receptor, we actually use it to inhibit the function of the NMDA receptor. So all these tools that I'm using, they are functionally inhibiting the NMDA receptor. So it's like a loss of function. But even if it's all loss of function, it may have many different outcomes. So this is what I really want to show you. And in the depression field, the hypothesis is that just a brief functional inhibition of the NMDA receptor that may drive long-lasting antidepressant efficacy. Good. So this brings us to the importance of timing. So NMDA receptor inhibiting compounds, they have acute effects that are induced by a compound in the brain targeting the NMDA receptor, but they also have long-lasting effects that last even if the compound has left the brain and make permanent changes to the brain. So when we study the acute effects, we may just inject the compound and look at how a rodent behaves in a box, but we may also do more challenging tasks. So we can look at balance, for instance. We can challenge with inducing seizures, or look at brain waves with EEG. This is good for the acute effects. When we study the prolonged effects, we can take out the brain and look at brain slices at many different times after one dose of the compound. And there's also a behavioral test where rats or mice swim in a pool of water, and this may actually also be affected by just one dose of the compounds. So what I will tell you about today is the effect of single dosing of one of these compounds. So if we take the basal locomotor activity first, all of our studies that I'm going to show here in the presentation are done in rats, humans. So what we do here is just taking a rat, injecting a compound, and then put it in a box, watching what's happening. In these boxes, there are photo beams, so movements of the rats are counted. And this is what you can see here in these figures. You see the count of movement. And is that a question or no? Okay. So when we dose different doses of ketamine that blocks the pore, then the high dose of the highest dose we tested would induce hyperactivity. And this would also be the case that you had actually long-lasting increases in activity levels by a compound that selectively inhibit the NR2B receptor subunit. But when modulating the glycine side, it did not affect motor behavior. And of course, the effects of ketamine and the MK0657 compound look very different, but that has also to do with the time the compound is in the brain. So it's actually mimicking the exposure of compound in the brain. So we can also challenge the rats by putting them on a wooden beam and ask them to to balance and cross the beam. And we did this also for the 3 types of compound. Ketamine at a dose that did not affect basal locomotor levels seriously disabled the rats so that they could not balance. So what you see in the gray column is how many times the rats slip when it's trying to pass the beam, and sometimes they even fell off the wooden beam. They don't fall that far, I can tell you. And at the highest dose, they could not even walk. So that is seriously affecting the balance of the rats. On the other hand, the NR2B subunit blocking agent did not have any effect on balance. If anything, these rats, they seem to be more alert, and they were faster to cross the beam. With the glycine side partial agonist, the D-cycloserine, they seem to be more relaxed, slightly sedated at the high dose, but they did pretty well even at the highest dose, where they got so contrary to the NR2B NAM, they were more sedated, whereas the 2B NAM made them more alert. Yes. So we also often test compounds like this on the protective effect on induced seizures. So there's a test called the maximal electroshock threshold test, where you test the anticonvulsant efficacy of a compound. And this is very sensitive to NMDA receptor inhibition. So even if GRIN patients may have epilepsy, the acute effect of blocking the NMDA receptor is actually protective towards convulsion. So here both ketamine and D-cycloserine. So this is for ketamine dose response. So the higher the dose of ketamine, the more it protects against seizures. And here it's a time series, so it shows how the effect wears off with time. Over here we have the D-cycloserine, so there's also a dose response effect of D-cycloserine, and that wears off but slower. So by inhibiting the NMDA receptor, we protect against convulsions acutely, and this is only while the compound is on the target. So this brings us to EEG. So this is it's more time consuming to construct these experiments. You have to go through surgery with these rats. We implant electrodes in the brain. So I'm trying to show you with this primitive movie how we put a screw in the skull to record EEG, or ECOG it's also called here. And then we also put some electrodes deep into the brain to record the activity. And this is of course not how it really looks, but it's to symbolize what's going on. So here are pyramidal neurons firing action potentials, and this is recorded by the electrodes, and it's acquisition by our acquisition system. And these signals we treat in different ways, filtered, to either get the activity of one single neuron or populations, so of thousands of neurons. And we try to look into how the oscillatory activity at different wavelengths are change with time. And normally it's quite stable. So this is what we do. We construct heat plots where blue is low activity and red is high amplitude activity. But then with if we give a compound a challenge, then we see changes in the different frequency bands. So you have, for instance, more fast oscillations and less slow oscillations. And we normally baseline normalize this to make the effects even clearer. So this movie was made by one of my former colleagues who was very nice to help us out here. So these are examples of these heat plots where we compare ketamine and D-cycloserine and another compound called DOI. It's actually closely related to LSD. And as you see, it's like they have different fingerprints, these compounds. But what's in common is that they all induce high frequency oscillations. That's the HFO. So these are fast oscillations around 150 oscillations per second. What is different is that, for instance, ketamine is increasing gamma oscillations. Those are from 30 to 60 hertz, and it's inhibiting lower frequency oscillations between 10 and 20 hertz. So this here you can clearly differentiate between ketamine effects and D-cycloserine effects. So here I'll try to put the three different types of modulation against each other. So because we put an accelerometer on the head of the rats, we also get a activity readout from our EEG experiments. So we always have a baseline of half an hour, then we inject compound, and that's always exciting the rats a little bit, and they'll run around no matter if they get a vehicle like a placebo injection or they get a real compound. But ketamine only has a weak effect on locomotion, whereas the NR2B NAM inhibitor through NR2B really increased the locomotion of the rats. D-cycloserine did not do much, if anything, slightly inhibit the activity. So then I also I tried to quantify the low beta inhibition that you saw with the ketamine, the blue area in the heat plot. So green here is ketamine and blue is a vehicle injection. So ketamine clearly inhibits these beta oscillations. Unfortunately, this is actually a bit difficult to pick up in the NR2B animals, because this is only something that we record when the rats are sitting still, and the rats were really not very quiet after this injection. So the statistics are not very great here, although that I actually think that there's an inhibition of beta oscillations also with the NR2B compound. D-cycloserine had no effect. If we then look at the high frequency oscillations, all three compounds increase that. So you can use EEG to differentiate between different types of modulation of the acute effect at the NMDA receptor. So I'm just trying to sum it up here. So ketamine and the NR2B compound inhibits beta, D-cycloserine does not. Ketamine increased gamma, but the two other compounds didn't really significantly do that. But they all increased the higher frequency oscillations. So there are differences in the lower frequency bands, but all the compounds share the increase in high frequency oscillations. And these effects are also following the exposure of compound in the brain. So it's acute effects. So now we turn to the long-lasting effects, prolonged effects. So this is what you can measure after the compound has been washed out. And often we look at 24 hours after a single dose. So this type of experiment is a hippocampal rat brain slice. So what we've done is that we inject rats with compound. Then we leave them in their cages for 24 hours. And then we take the brain out, slice it, and we can then in a multi-electrode array system stimulate one area of the brain and record in another area of the brain. And here we've shown that ketamine is increasing excitability. So what we record is a field potential, and this is excited by ketamine. So less current is enough to induce a strong signal here. So ketamine, 24 hours after ketamine injection, where ketamine is completely out of the body, something has changed in the hippocampus. And this we also record with D-cycloserine. So both of these compounds, they can increase these currents, and they are mediated by AMPA receptors. So this is the AMPA receptor somehow taking over because the NMDA receptor has been inhibited just for a short while. Then I mentioned this SWIN test. It's called the PROSALT SWIN test. It's a rodent behavioral test that has been used for many years to evaluate the effect of antidepressant drugs. So it's not like the rat is depressed, but it's stressed by having to swim in a bowl of water, and a coping strategy to this is to just float and relax. But antidepressants have this effect that they make the rats swim for longer. And we also tested our compounds here because it's a good way to show that something has changed in the brain. And ketamine is known to have an effect 1 day after treatment. So again, you dose, leave the rats, and then test them 24 hours later. And both ketamine and D-cycloserine would decrease the immobility 24 hours after dosing that. So that means they swim more. The contract research organization that did these experiments for us also tested S-ketamine. So that's one enantiomer of ketamine that is very potent. And there they could actually see an effect that lasted at least 7 days after one injection of ketamine. So just inhibiting the NMDA receptor for a brief period, so less than only a few hours, is enough to induce long-lasting changes in the brain. So this is a very densely populated table. It's to show how the site of action is important. So I list the different types of assays that we have used, and to help you, I put some boxes around these numbers. So what is in common for all of the compounds that inhibit the activity of the NMDA receptor is that they protect in seizure assays, and they also induce high-frequency oscillations, and then they have long-lasting effects. And of course, I have not tested the NR2B NAMs here, so I can't really for sure promise you that would also happen. But I've seen other data on this. So then, what is the difference? So only the NR2B compounds and the pore blocking agents increase locomotor activity. This is what you have here. So resting state, EEG, LMA, that's locomotor activity, MOD-R, that's motor activity as well. So that is increased by these compounds. and then they also tend to decrease the slower oscillations in the EEG. What is special for NR2B negative modulators is that they have absolutely no negative effect on the balance in the beam walk, whereas ketamine was very disruptive, and at high doses, D-cyclocerin was also sedative here. And what is specific for the glycine side modulation is that no matter how high we dose this compound, it would never induce hyperactivity. So how does this translate into human behavior or EEG for that sake? So we asked some colleagues at King's College in London to run an experiment for us where they tested 2 doses of D-cyclocerin, 250 milligrams and 1 gram of D-cyclocerin against half a milligram of ketamine and placebo. So what they did was to place a cap with an EEG cap on the healthy volunteers, and then record EEG before dosing and at the time where both compounds were present in the brain, and then at a later time point where ketamine had been washed out of the brain, but D-cyclocerin would still be present. And then they also did a lot of other tests, for instance, to assess the effect on dissociative side effects. And let's start with the psychotomimetic effects in healthy volunteers. So these 2 graphs show the effect of D-cyclocerin before, when D-cyclocerin is at its highest concentration in the brain, and then at a later time point, there's no effect on the psychotomimetic scores. Ketamine has a really high impact at when it's in the brain, but it is quickly flushed out of the brain. Placebo did not really have a strong effect. So again, here we can differentiate between blocking the pore and targeting the glycine side at the NMDA receptor. So then the EEG is, I think, really interesting because that really show a great translation between the rat and the human. So this orange or reddish line here is ketamine, and it inhibits the lower frequency 10 to 20 up to 30 hertz oscillations, just as in the rat. But placebo or D-cyclocerin had no effect here, just as in the rat. Ketamine is inducing gamma, and that was also strong in the rat. D-cyclocerin is not doing that. But again, ketamine and D-cyclocerin increase the higher frequency oscillations. So this supports that D-cyclocerin and ketamine elicit similar but not identical neurobiological effects, and this is something that we can record in humans. Yeah. So let's return to the questions from the beginning. So you know why I was studying the NMDA receptor, because I was fascinated by these rapid-acting antidepressant effects of NMDA receptor antagonists. And I tried to show why I think the site of action is so important. So loss of function, that can be many different things depending on where you hit at the receptor. And then, of course, the timing is of importance. What I have studied is only a brief inhibition of the NMDA receptor, and this, of course, does not compare to a genetic deficit. I think for the EEG, there's a very good translation to humans. So this can also, of course, be a back translation. So if you have genetic models of GRIN mutations, then perhaps there's a good chance that they would also translate. This is my belief. And then the big question is whether this really translates, and I guess this is what we should discuss afterwards. So that was what I had to say. I'd like to acknowledge all my great colleagues at Lundbeck, the people helping in the lab, PhD students, and department heads, and many more, and the people at King's College who did this really, really important clinical trial. And then our friends in Barcelona, Francesc, Artigas, and Pau, who were supervising Maria Amat Forrester, a great PhD student that is not a PhD student anymore, but they're working here at Lundbeck. So that was it. Thank you very much.
Okay, thank you very much, Kьяртан. It was really, really interesting. Now, because we are running a little bit past time, I will quickly introduce our next speaker, Jenny-Li. I hope you're ready, because I can see you.
Yes. Can you hear me?
Yes, we can hear you. As I previously mentioned, Jenny-Li, she's a mom of a child with a different genetic disorder, but nonetheless, maybe even more complex than our GRIN children. So she has a wealth of knowledge. And over to you, Jenny.
Thanks. Let's see. So I'm going to start with a small presentation about me, because I think that has some relevance to my standpoint. And my name is Jenny-Li Orselll, and most of you know me as a very frequent poster at the GRIN research community at Facebook. And some others of you know that I'm the parent of a little girl with SAPB2 syndrome. Here you can see her, and two of the features of SAPB2 syndrome, a broad smile and some odd-shaped teeth. Otherwise, the SAPB2 syndrome is pretty similar to GRIN disorders, and there is a link because the SAPB2 gene is expressed in the nucleus of pyramidal cells of the brain, and it regulates a lot of other genes, and probably among them the GRIN genes, but we don't know how yet. And I'm very obviously passionate about translating science to treatments. And my little girl here, she is the reason for that. And just for the record, I don't have any kind of medical background. Actually, my story started 3 years ago when we got this diagnosis, and it's really, really rare. I think there were less than 200 people diagnosed with this when we got her diagnosis. And actually the same week I left my job and started reading up on the literature, because I wanted to learn as much as I could about this very rare syndrome. And it took me about 2 months until I found the link to the GRIN genes, and I joined the GRIN community. A year later, I thought that it was time to get back to work, and lo and behold, I got a position at the Department of Clinical Neuroscience at my country's top medical university. And after that, I decided to start a SAPB2 research community, just like the one you have for GRIN. And today we have 350 members, so we're still a smaller sibling to your research community. And I also started to contact researchers. In the beginning, I had the questions, but after a while, people started to ask me things. Half a year ago, I was asked to co-author my first scientific article on EEG abnormalities in the syndrome, and I actually have another article in the pipeline. So as the cherry on the top, today I'm here as a speaker at the 3rd European GRIN Conference. And it's been a very exciting journey, of course, but most important is, of course, that I've been able to use insights from my own research to raise the quality of life for my family, My kids means the world to me, and they are my motivation. So here's my presentation, and it's called Beyond the NMDA Receptor. And why is that? Well, we've been focusing a lot on this lately, and this is the NMDA receptor, and you can see the flow of ions through it and a couple of substances that act directly on it. And it is a great approach. I mean, once we find drugs that can modulate this flow through the receptor, we will be able to help many GRIN individuals. But there is certainly more going on in GRIN than just this receptor and the flow through it. And more, there are many ways to look for solutions. We will get some solutions if we look at GRIN disorders this way, as a receptor. But we can look at what the receptor does at the synapse, and we will get other solutions. We can look at whole organs and how they interact with others when there are mutations. We can look from the outside, like a physician, and see what kind of solutions can we find for what we see. We can look at behaviors and try to treat those. We could look at EEG abnormalities and try to treat those. We could take the genetic approach, and our solutions will probably be gene editing. We can look at cells in vitro, or we can look at animal models. And depending on how we look at GRIN disorders, we will get different solutions. Another thing I find really cool is that almost all disorders that affect cognition, memory, and behavior, they share some mechanisms. And this is regardless if the origin was genetic or if it was acquired from an injury or caused by an immune hit. And what this means is that rare disorders like GRIN disorders or SAPP2 syndrome are very similar to other disorders. And it means that treatments for other disorders may apply to GRIN disorders. And we don't have to do everything from scratch. We can save money and time this way. And this is the foundation of a rare parent strategy, because a rare parent seldom has research budget or animal models or a lab. We have to be very smart. So once we find how our disorder relates to these common mechanisms I'm talking about, we can apply a divide and conquer strategy to manage symptoms. And so what are these common mechanisms I'm talking about in brain disorders? Well, I show you a few. And when you look at them from the top, it will look like a scrambled jigsaw of pathological puzzle pieces. So we have disturbed EI balance, oxidative stress, blood-brain barrier disruption, neuroinflammation, poor myelination, mitochondrial dysfunction, and so on and so on. There are very many of them. And the great question here is, how do they all fit together? In fact, I don't believe there is any person in the world who would be able to fully answer that. And there's a lot of scientists who are occupied by trying to find out how they fit together. But there is some consensus on how some of these puzzle pieces fit together. And today I'm going to try and tell you a little bit about these connections. So it's going to be a very brief overview. It's going to be overly simplified, but hopefully relevant for GRIN disorders. And I wanted to start with something we know. We know that the GRIN proteins build up the NMDA receptor, and we know that mutations will cause the NMDA receptor to dysfunction or be disactivated. And that is our first mechanism. In GRIN disorders, we know it's a dysfunction, in other disorders, it's more about disactivation. And the NMDA receptor mediates excitatory signals, but they are also to some extent involved in inhibition. And thus the NMDA receptor dysfunction will affect the EI balance. And that is our second mechanism. And the E is, of course, for excitation, which in layman's terms would be something like signaling or messaging or information. And the I is for inhibition. And in layman's terms, that would be noise cancelling. So here you can see a signal. We need excitation for the signal, and we need inhibition to cancel every noise. And we need both of them to be in balance for a clear signal to go through and for proper cognition. So this EI imbalance is a well-recognized cause of epilepsy. And excitotoxicity is a lighter version, I would say, than epilepsy. And we see seizures in many GRIN-affected individuals. And that's independently if they have gain of function or loss of function mutations. So the NMDA receptor is triggered by glutamate. And in other disorders, too much glutamate is a problem. But is there excessive glutamate in GRIN disorders? We don't know. We can only speculate. We might want less glutamate if we are dealing with a hyperactive receptor. And maybe more glutamate is released to trigger receptors that are underactive or even missing. But we don't know. So the next mechanism I'm going to talk about is oxidative stress, and I'm sure you have heard about it before. It's another kind of imbalance. This time, on the one side, we have reactive oxygen species, and here they are represented by a superoxide anion. It's a free radical, and you can see it by that little red dot, which is an unpaired electron, which makes this molecule very unstable and highly reactive. And on the other hand, we have our antioxidant defenses. And this time I'm representing that with a glutathione molecule. And glutathione is our main antioxidant substance in our bodies. So oxidative stress happens when the amount of reactive oxygen species is higher than an organism's ability to neutralize them via its antioxidant defenses. And just for the record, there are reactive nitrogen species too. I'm not going to talk about them today, but they are very similar. So these reactive oxygen species in excess are mostly bad news because they can do a lot of damage to a cell and its components. It can damage fats through lipid peroxidation. It can damage proteins, and if a reactive oxygen species can travel down to the nucleus and cause base damage or even strand breaks in your DNA. And that's thought as one cause of cancer. So it's really nothing you want for your kid. Oxidative stress happens in all brain disorders. In fact, it happens in all brains too, to some extent, but it's contributing to almost all brain disorders that I know. So what does the literature say about GRIN disorders? Well, we don't have anything on GRIN disorders, but we can look at something that is pretty similar. Here is one article. It says, activation of neuronal NMDA receptors induces superoxide-mediated oxidative stress in neighboring neurons and astrocytes. So this could be relevant if you have a gain-of-function mutation, because then you would perhaps have more oxidative stress. And here's another one. It says synaptic NMDA receptor activity boosts intrinsic antioxidant defenses. Well, that sounds good, unless you have a loss of function mutation, because maybe you will have less antioxidant defenses then. Who knows? And the third one, does the redox balance of the brain, acting in no small part via glutathione, controls NMDA receptor activity? So it looks like there is a reciprocal relationship here between the NMDA receptor and oxidative stress. And I'm going to add all these relationships to our figure here at the left. Yikes, what is that? Oh, I'm sorry. That is a mitochondrion, and it is our main suspect for production of reactive oxygen species. So let's add mitochondrial dysfunction to our mechanism. So the primary task for mitochondria is to supply our cells with energy. And they do that through a process that is called oxidative phosphorylation. And ATP is the energy unit that our body uses. And this oxidative phosphorylation can be disturbed when too much calcium flows into a cell, because mitochondria act like sponges for calcium. So think of gain of function mutations or during seizures. And the oxidative phosphorylation can also be disturbed by oxidative stress. And when this happens, mitochondria will start to produce a lot of reactive oxygen species themselves. So we get another couple of relationships here at the left. And note that mitochondrial dysfunction is also a cause of epilepsy. What does the literature say? One article says excessive activation of NMDA receptors in the pathogenesis of multiple peripheral organs via mitochondrial dysfunction, oxidative stress, and inflammation. And here's another one. Mitochondrial dysfunction is a primary event in glutamate neurotoxicity. And this one was on the NMDA receptors. So yes, it looks relevant for us. So I'm going to add another mechanism. It's blood-brain barrier disruption. So what is that? Well, the blood-brain barrier is found at the lining of these thin blood vessels in the brain. And it's made of a layer of endothelial cells. If you look at the right, it's the violet cells lining the blood vessel. And they are connected by tight junctions. And these endothelial cells are the same type that you will find in your guts. And the blood-brain barrier keeps pathogens, immune cells from the rest of the body, and other large molecules, it could be drug molecules, for example, out of our brains. And it also regulates the inflow of glucose and ketones. So, is the blood-brain barrier relevant for brain disorders? Here's one article saying non-ionotropic action of endothelial NMDA receptors on blood-brain barrier permeability. And the non-ionotropic word here means that this is regardless of any flow of ions through the NMDA receptor. So yes, relevant. Let's add an arrow for that to our figure. So seizures is probably most well-known cause for acute blood-brain barrier disruption. And this is linked to NMDA receptor signaling. If you have chronic epilepsy, it could result in a leaky blood-brain barrier. Oxidative stress is also linked to blood-brain barrier disruption. And blood-brain barrier disruption is a part of how myelin gets damaged, at least in one disorder called multiple sclerosis. And we need the space, so I'm gonna hide that part for a while. Most important, blood-brain barrier disruption will result in neuroinflammatory processes and increased excitability and risk for new seizures. So what does the literature say about this neuroinflammation? A direct relation between NMDA receptor constant activation and inflammatory cascade activation exists. Chronic brain inflammation leads to a decline in hippocampal NMDA1 receptors. Here we show that peripheral inflammation increased the expression of NMDA, NR2B receptors, and NR2B receptor-mediated synaptic currents in the anterior cingulate cortex. That's a part of the brain. So, neuroinflammation. Acutely, it will protect the brain. But when it's sustained, it will cause sickness behavior. It will cause cognitive decline and, in the long term, neurodegeneration. And the brain has its own specialized immune cells that you can't find anywhere else. And they're called microglia. Here's a microglial cell. And when it's activated, it will release pro-inflammatory molecules. And you've heard about them before, cytokines. You've probably heard about them during this pandemic, but then it's other types of immune cells that shares these cytokines in our lungs. And the issue here is that insults such as early life seizures can predispose these microglia to become primed or hyper-responsive. And the thing is that microglia will stay primed for a very, very long time, and they live for a long time. They live for years. And once they are primed, they will react to any kind of inflammatory signal. And I'm using quotes here around the word inflammatory because most of us will think about inflammation as, I don't know, bacterial inflammation. But in the brain, there's also sterile inflammation, which can be caused by a lot of things like epilepsy, oxidative stress, mitochondrial fragmentation, or myelin debris. Even social stress can activate microglia, at least in mice. And microglia will also react to signals that come from the rest of our bodies, such as virus infections, allergies, food intolerances, gut issues, and even tooth eruption. And all of this happens frequently in small children. And the thing is, this doesn't have to happen through a blood-brain barrier disruption. There are other ways for these inflammatory signals to go to the brain, via the vagus nerve, for example. Neuroinflammation is a somewhat self-perpetuating process because activated microglia will degrade the blood-brain barrier, and they will recruit immune cells from the rest of the body that doesn't belong to the brain. And they will activate other microglia and kill brain cells. And all of this will further trigger inflammation. Neuroinflammation will also get more glutamate released, and it will inhibit reuptake by astrocytes. So more glutamate. It will also push more calcium through the NMDA receptors. I think that is through an effect on the NR2B subunit. I can't remember right now. And it will reduce GABA inhibition by acting on chloride transporters. And it will get more reactive oxygen species released. So when we look at this picture, we can almost see there's a vortex or a vicious cycle of mechanisms that enhance each other and trigger each other. So before I stop this 15 minutes lesson on mechanism in brain disorders, I wanted to add a little bit on behavior at the end, because behaviors is what we see as parents at home, and we all always wonder why is that. So neuroinflammation is in itself will cause sickness behavior, but it will also cause something called tryptophan metabolism to be skewed. And tryptophan goes through two pathways, and one pathway is going to serotonin in the brain. And when it gets skewed, we will get brain serotonin depletion, which will cause behavioral abnormalities. And this has been tested in autism, and what we see then is whirling, flapping, hand flapping, pacing, banging and hitting self, rocking and toe walking. And I'm sure there are parents here who have seen some of these behaviors at home. It will also skew the kynurenine pathway, and that one is very interesting because some of the metabolites there are agonists or antagonists on the NMDA receptor. And of course, the NMDA receptor is linked to the dopamine 1 receptor, so we will see dopamine issues too. Wow, that was a lot to take in on 15 minutes. And I'm afraid there are parents out there who are not thinking, wow, about this, but oh my God, is all of this happening to our kids? What are we gonna do? And I can assure you, I wouldn't tell you about this if it was bad news, but actually there are many possibilities for us to act on this. And I'll show you how. Today, we have our clever researchers who are looking at GRIN disorders from this viewpoint. And they are going to find some really nice treatments for our kids. But as a parent, when I look at this box, I feel like the number of possible treatments is not that big. So what I think is that the rest of us, in the meantime, maybe could take a broader view on the issues, and maybe we'll find other treatments that are applicable to GRIN disorders, because there are so many solutions out there that have been used or trialed for these mechanisms in other disorders. And here's just a few ones that I find promising or interesting, and several of them have been trialed on children. And I even think we have people in the community who have tried some of these. So by looking at GRIN disorders this way, we can understand why the treatments we've tried so far may not suffice. One example would be the anti-epileptic drugs that do not always work for GRIN disorders. Maybe we need to address mitochondria, inflammation, or oxidative stress, and not just the EI balance. And we will be able to find new solutions that solve certain symptoms. And I'm not talking about cures, but how to raise quality of life as soon as possible for our kids and for our families. And some of these solutions will have an impact on several mechanisms at once. I'm a fan of flavonoids because they are so broadly acting. And many solutions will amplify each other. So it's really a divide and conquer strategy, and it's useful for anyone. I believe any parent can read up on these mechanisms, and you don't need to be a researcher for that. You might become one, though. And the greatest answer may not lie where we expect to find them. Just because we call GRIN disorders neurological today, we don't know about the metabolic or immunologic impact yet. Until we know which mechanisms are the most important ones, we need people to turn every stone out there. And maybe you can turn one stone for the team. Thank you for listening. My acknowledgement goes to parents, bloggers, inventors, biohackers, and researchers who taught me stuff that neither school nor literature can. And you can reach out to me either on Facebook or on my email below. Thank you.
Thank you, Jenny-Li. This was truly awesome. Maybe we should do this like on a regular basis with you. Like have this Jenny-Li corner in our Facebook GRIN research group, but we'll talk about that later. Okay, I think we have like 5 minutes for questions. And Sandra, would you please take over the question part, because I haven't been following the Q&A.
Yes, actually, I was trying to look for questions that were pertaining to what we were discussing. Oh, maybe I'll read this one, but I think we can leave it open for anybody to answer. I just wanted to check that ketamine, etc., are inhibiting, so therefore are appropriate for gain of function GRIN patients.
Yes.
I agree, but we just have to remember that there are so many side effects that are unwanted with ketamine, and the effect is very short-lasting. So it's not a good compound if you want to treat something that is long-lasting. The ketamine effect wears off in an hour.
Yeah, well, then I'm gonna keep on reading a few, and whoever of the panelists are here want to intervene, that would be great. There's another comment here. My 8-year-old grandson has a GRIN 1 gain of function. His only seizures are febrile seizures. Is there a relationship to epilepsy? I guess the epilepsy not caused by the febrile seizures. Besides memantine, is there any other protocol currently being used for the treatment of gain of function? No takers? We can leave that one for the round table discussion then.
Sandra, there's one specifically for Kьяртан. I think it's from Chavi. Maybe we should start with that
Okay.
And leave the rest for the roundtable discussion, maybe.
Mm-hmm.
You want to read that?
Ah. Sorry. Yes, Kьяртан, thank you for your great talk. Several GRIN mouse models are currently available and many others are in progress. Does Lundbeck consider the possibility to adapt EEG recording devices for the mouse models? Alternatively, are you considering the possibility to create GRIN rat models? As far as I know, the GRIN 2A het rat is available, which are particularly interesting in terms of neuronal and behavioral phenotypes. Thank you.
Yes, so it's not a problem to run EEG in mice. We do that at Lundbeck. And mice are more accessible for mutational models, but there are also some rat models. We don't generate models ourselves, but if we can access them and it can help us, we use them. It's just the mouse is so much smaller, so you really have to be careful how much weight you put on the head. But it's doable both to put screws in the skull and also depth electrodes. We can do that.
It looks very sci-fi, that.
It's just a cartoon, but that's kind of how it looks.
Well, thank you to both of you, Jenny-Li. Also, thank you very much. It was a great presentation. I think we all enjoy it, and we might have some more questions later on for both of you.